Hi, Namaste. Good evening to all. Myself, Murali.P.G. Today I am going to take a YouTube class on molecular basis of inheritance. Okay, YouTube class on molecular basis of inheritance is the chapter. Under this Unacademy, here we introduced plus subscription courses for the student point of view under Karnataka CET 2021 Aspirants. So best plan for that students is 12-month plan, then a 6-month plan and 1-month plan will be there, and best premium content at affordable price, structural live courses, dedicated live doubt clearing sessions, quizzes at regular intervals for self-analysis, batches for complete preparation, test series for DPPs, full-length mock tests etc. with the analysis, and single subscription for unlimited access will be there. And use my referral code Murali.P.G 85 3057 and get 10% off regarding with this price value. So Unacademy is one of the good platform for your achievement in the case of entire oncoming Karnataka CET 2021. Secondly, we have with another important class, that is the comprehensive batch, where it is complete, where in that batch or in that course complete syllabus is going to be covered, and tips and tricks by top educators, topic wise practice questions will be there, and full length mock test will going to be carried out. So here also it is one of the best plan, because it is here complete syllabus is going to be covered with the tips and tricks by top educators, plus educators will be going to take. Myself, I am taking this class from last 2 months, and Murali.P.G 85 3057 is my user code and use code, so you will get 10% off if you use my this particular code, okay, during subscription. So as a result, you will get 10% off. And the third one is a chartered batch for Karnataka CET 2021, where PCM, PCMB and PCB batch will be there, and started on 15th of October, and enroll your name for this course also, during which you can get the 10% off if you use my referral code Murali.P.G 85 3057. And the last one, it is a daily T20 free practice test for Karnataka CET, started on November 2nd. Enroll for the test and attempt it live. Let's crack KCET together. Okay, this is also going to be helpful to you to achieve a good goal in your oncoming Karnataka CET exam. So this is about the brief about promotion slides regarding with our Unacademy. So now let us start our discussion of questions, my dear friends. Okay. If you have any doubt, you can ask. Okay. If you have any doubt, you can ask with me. So this is the YouTube class regarding with our molecular basis of inheritance. Molecular basis of inheritance refers to your second chapter of the genetics. Molecular basis of inheritance. Okay, got it? So in that first question, select the correct statement. Select the correct statement. Here, R strain bacteria has a mucus coat. S strain is virulent. R strain is virulent, and both S and R strains are virulent. So which is the right one? So here the answer for this question, it is S strain is virulent. The smooth strain is virulent because it is having the mucus coating, which will going to cause streptococcus. It is going to cause pneumonia by this streptococcus pneumoniae S strain. So one of the very important experiment in the case of molecular biology, and this experiment will going to give the transformation principle, isn't it? Yeah, Griffith transformation principle. So the bacteria streptococcus pneumoniae will going to have the S strain will going to have pneumonia fever in the mice, isn't it? So Chetan Rathod, welcome to the class. Good evening. So the second question is who proved that DNA is basic genetic material? Who given the proof for DNA is basic genetic material? Answer is B, that is Hershey and Chase experiment. Hershey and Chase did the experiment and came to know that DNA is a basic genetic material. So the pH is basic with respect to DNA. Then the third question, if you have any doubt you can ask, okay. The third question, biochemicals purified by Avery from the heat killed S cells are which one? So Avery you will going to do the continuation of Griffith experiment, and he collected some of the biochemicals from the heat killed S strain of Streptococcus pneumoniae, which is that thing. So answer for this question is D, all the above, because we got protein, RNA and DNA. So answer is D for third question. Manoj, welcome to the class. Good evening. So we are in third question. All the above is the right answer. Okay. All the above is the right answer. Got it. Next, fourth one. The main category of mutation among the following is which one? The main category of mutation in the following thing: genetic mutation, heterosis, somatic mutation, none of these. So which is the main category? It is the A, genetic mutation. Got it? The first one, A, is the genetic mutation. It is the main category of mutation among the following. So the option is B for that one. Okay. Sorry, A is the right answer for fourth one. Next, the fifth one. One of the following which causes mutation is caused by mutagens, isn't it? Mutagens are the molecules, substances responsible for causing of mutation, which is that one. Now the answer for this one is B, X-ray. Prolonged exposure of body into X-rays will going to cause the mutation. Okay, so mutation is caused by mutagens, and prolonged exposure of body into these mutagens like X-rays, UV rays, etc. So in that ionizing radiation and non-ionizing radiations are there under mutations. Okay, so right answer is B for fifth one. Yes, Manohar, welcome to the class. Good evening. Yes, X-rays. Then sixth question, number 6. Lac operon is an example of which type? In the operon we can observe inducible operon, repressible, overlapping genes, none of this. So with respect to our syllabus, the Lac operon, it is a inducible type of operon. Inducible type of operon, that is the Lac operon model. Okay. Sorry. The next question number 7. Which of the following gene is not a structural gene in the Lac operon? Structural genes refers to they are responsible for the production of proteins or expression, isn't it? So from the structural gene, mRNA is going to be produced. So which is the gene which is not coming under structural gene? So for the seventh one, B is the right answer. So it is LAC-I. LAC-I is for repressor protein, isn't it? Repressor protein. LAC-A, LAC-Z, LAC-Y. It is the three important structural genes present in the Lac operon, whereas LAC-I is not observed in that structural gene complex. Okay. So right answer is B for 7th one. Then 8th one. Enzyme needed for transportation of lactose into the cell is which one? Enzyme needed for transportation of lactose into the cells. Into the cells means inside the cell of that E. coli or Escherichia coli. Which is that one? So here galactosidase, transacetylase, lactase and permease. So 8th one, it is the permease. Permease will going to give permeability for the entry of this lactose into the cell, when the lactose is the chief energy source in the media of that E. coli. That is permease. Okay. So this permease enzyme is produced from the structural gene, out of 3 structural genes. So answer is C for question number 8. Next, question number 9. DNA fingerprinting was developed by whom? Who developed DNA fingerprinting technology? DNA fingerprinting ani yaru introduce ಮಾಡ್ತಾರೆ? Who introduced that one? Corona means corona refers to scientist name. It's not a coronavirus. Okay. Corona, James Watson, Alec Jeffreys and Messelson. Who introduced this one? This is the Alec Jeffreys. So Alec Jeffrey introduced the process of DNA fingerprinting or DNA profiling, isn't it? DNA profiling of the person. So one of the important phenomena which is needed, or one of the important achievement in the field of molecular biology, and now we are using this one in forensic science also, isn't it? So Alec Jeffreys, the father of DNA fingerprinting. Then each individual has a unique DNA fingerprint, as individuals relies on what? So particular every individual organism, if you take human being, we are with a particular type of unique DNA fingerprint, as individuals relies on what? Location of mini satellites on chromosome, number of mini satellites on chromosome, size of mini satellites on chromosome, all the above. So it is all the above. Depending upon this one, we can have the particular type of fingerprint. So location of mini satellite molecule on chromosome of that particular individual, number of mini satellites on chromosome of that particular individual, and size. So location, number and size are very important in the detection of that particular DNA fingerprinting matching. So answer is D for 10th question. Okay. Yes, if you have any doubt, you can ask my dear students. Okay, next question number 11. Human genome consists of approximately how much number or how many number of base pairs? What about the base pairs observed in the case of human genetic material? So 11th one, it is C, 3 into 10 to the power 9 base pair. It is the approximate genome, total number of genetic material, total amount of genetic material present in human being with respect to their genome. It is the 3 into 10 to the power 9 base pair. So answer is 11th one, it is C. So Rakesh, welcome to the class. Good evening. And 12th question. If you have any doubt or if you want any clarification, please ask. Okay. Reason for discontinuous synthesis of DNA in one strand is what? What is the reason behind that one? So here for 12th question, C is the right answer, but here one mistake is there. The arrow mark should be from 5' to 3'. Okay, here the arrow mark should from 5' to 3' here in the third option. So right answer is C, DNA dependent DNA polymerase catalyzes polymerization only in one direction, that is 5' to 3 prime direction here. Okay, 5 prime to 3 prime direction. Here 5 prime to 3 prime direction. Okay, I made the correction here. It should be from 5 prime to 3 prime direction. Okay, so answer is C is the right answer for question number 12. For question number 12, C is the right answer. Okay, 5 prime to 3 prime direction. C is the right answer for that question. Next, 13th one. Next question, 13th question. Read the statement and select the correct option. First statement says typical nucleosome contains 200 base pair of DNA helix. Densely packed and dark stained regions of chromatin is called as euchromatin. Loosely packed and lightly stained regions of chromatin is called heterochromatin. Which are the right and which are the wrong statements? So which are the right and which are the wrong statements? So 13th question. Here typical nucleosome contains 200 base pair of, yes, that is right. Densely packed and dark stained regions of chromatin is called euchromatin. Loosely packed and lightly stained regions of chromatin is called heterochromatin. So here a small change, okay, a small change, yes. The right statement is only first one. Only first one is correct. Okay, only first one is correct. Okay, so we are going to change that one here. Only first one which is right. So answer is A for this question number 13. Typical. But what about this second and third? It is densely packed and dark stained regions of chromatin is called heterochromatin, isn't it? Loosely packed and lightly stained regions of chromatin is called euchromatin. So vice versa. This is wrong, isn't it? So only A is the right thing. Only the first sentence is right. Then 14th one. Select the incorrect statement regarding DNA replication. Incorrect one. So for 14th one, A is the incorrect. DNA polymerase catalyzes polymerization in 3' to 5' direction. It is 5' to 3' direction. So that is the wrong statement or incorrect statement for 14th one, isn't it? 14th one, yes, got it. Rakesh, Manohar, Chetan, Manoj. So right answer is A for 14th question. 15th one, select the wrong statement with respect to DNA polymerase 1 enzyme. DNA polymerase 1. Here, yes, Manoj, for 15th one, select the wrong statement with respect to DNA polymerase 1. It is the main polymerizing enzyme of E. coli. No, it is not the main polymerizing enzyme of E. coli. Okay, it is a DNA polymerase 3. So in that DNA polymerase 1, 2, 3 is there. Okay, so right answer is A. It is not the main polymerase enzyme of E. coli. Next, structural genes in eukaryotes possess coding and non-coding sequences called 1 and 2 respectively. So structural genes in eukaryotic gene. In the eukaryotic cell, where the structural genes are present, which are with the coding and non-coding sequence. What is that coding sequence and non-coding sequence? So ಯಾವುದರ ಕೋಡಿಂಗ್ ಸೀಕ್ವೆನ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ, ಯಾವುದರ ನಾನ್ ಕೋಡಿಂಗ್ ಸೀಕ್ವೆನ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. So 16th one, right answer is coding sequence is called exon and non-coding sequence is called introns. So exons and introns, isn't it? So exon and intron. So coding sequence is called exon and non-coding sequences are called intron. So during the time of production of RNA, mRNA, after or before the processing, there is a removal of intron part, only exon parts are going to be rejoined, isn't it? Otherwise, what happens? Introns also going to be there in the mRNA. It is actually it's a negative aspect for the protein synthesis, isn't it? So introns are not needed. So only the exons are needed. Only the coding sequences are needed. So processing of mRNA will be there in the case of eukaryotic gene expression. But in the prokaryotic gene expression, intron exon concept is not there. Okay. So introns are removed by using spliceosomes, and exons are joined, and heteronuclear RNA is going to be processed, and matured mRNA is going to be there. So it is having tailing and capping phenomena ultimately, and then that processed mRNA will travel from nucleus into the cytoplasm, where protein synthesis will going to be happening. So right answer for 16th one, B. Then 17th one, 17th question. Here, select the wrong statement. 17th one. It is in prokaryotes, primary mRNA transcripts undergo splicing. It is a wrong statement, isn't it? Just now I said. Yes, Rakesh, Manoj, got it? Manohar. So it is in prokaryotes, the primary mRNA transcript undergoes splicing. In the prokaryotic, there is no splicing. There is no intron-exon concept. Only the exons are there. All are coding sequences in the case of prokaryote. Okay. In the case of prokaryotic gene expression, all the genes are exons, and it is the coding sequence. There is no splicing phenomena. There is no processing of mRNA. Okay. So wrong statement is C. Then mutations are responsible for what? It is variation in organism, increase in the population rate, maintaining the genetic continuity, and change in the environment. So it is the variation in organism. So we can observe variations. So variations will going to have the origin of new species, isn't it? So mutations are the raw material for evolution. Origin of a new species means evolve. So new organisms are evolved at the head. So this phenomena of mutation will going to help with respect to variation in organism. Mutations are the raw material for evolution. Okay. So answer for this one, it is A for 18th one. And next 19th question. 19th question. There are totally I think 30 questions are there. Yeah. 19th question. The base responsible for hot spots for spontaneous point mutation is which one? Hot spots for spontaneous point mutation means in the DNA sequence there is different bases, adenine, guanine, cytosine, like that. So which base is going to be considered as the hot spot for point mutation, a single base pair change? So which is that one? That is guanine. It is more susceptible for the spontaneous mutation. It is more susceptible for spontaneous mutation. So the right answer for this question is B for 19th one. Okay. So thank you, Rakesh. Yes, 19th one, that is B. Manoj, you are right. Good work. Okay. Next question number 20. Identify the incorrect statement. Incorrect. So incorrect one is average gene consists of 30,000 base pair. Earlier, before the Human Genome Project, the genetics says that it is 30,000. But after this Human Genome Project conclusion, it is not average gene consists of 30,000 base pair. Okay. So the incorrect statement is C. What about A? Chromosomes of Y has 231 genes. Least number of genes present in one chromosome is Y chromosome. Y chromosome consists of least number of genes. Less than 2% of genome codes for protein. Yes, the remaining things are repeated sequences, isn't it? Then human genome consists of 3164.7 million nucleotide bases. That is also right. So wrong statement is C for 20th one. 21st. The function of beta subunit of polymerase is what? Beta subunit of polymerase enzyme during the processing. So that is 21st question. B is the right answer. Catalytic binding. Catalytic binding process. Okay. Catalytic binding process. So 21st B is the right answer. Yes, Manu, you are right. 30,000 B square for the previous question. And next question. Which of the factors, sigma factors, is heat stable? Heat stable during the time of top polymerase action, isn't it? Thermus aquaticus. In the case of PCR, it is going to be there. So which of the factor, that is the sigma factor, is heat stable? So 22nd question, it is D, 32. Okay, sigma 32 is having heat stable property. 22nd, D is the right answer. Just a minute. So D is the right answer for 22nd. And 23rd, yes, sigma 32, you are right. Yes, Shishika, good evening. Welcome to the class. My dear students, on the next YouTube class will be on 23rd, okay, on 23rd of this month, and topic is strategies for enhancement in food production. Strategies for enhancement in food production. That is the next topic under my YouTube class. Okay, that is on 23rd of this month. 23rd of this month. Time is 9 o'clock, 9 p.m. Okay. So next question number 23. The mRNA codon of valine is which one? So valine is replaced by glutamic acid, isn't it? That will going to cause one particular disorder. Which is that one? Valine is going to be replaced by glutamic acid in the 6th position of beta chain of hemoglobin. Beta chain of hemoglobin in the 6th position. Valine is replaced by glutamic acid and going to cause one particular disease, which is that one. Yes, here mRNA codon for of valine is which one? Answer is GUC. Is it GUC? So which is that one? For the question number 23rd. So mRNA codon of valine is GUC. Answer is A. So valine is replaced by glutamic acid. Research in which disorder? Genetic disorder. Sickle cell anemia, isn't it? Sickle cell anemia is the answer for that question, additional question. And next, for 24. True replication of DNA is due to what? It is 24. It is C, complementary base pairing rule. Actual replication of DNA is based on what? Main principle behind that one. How the actual copy is going to be produced. Replicas are going to be produced. Xerox copies are going to be produced because of complementary base pairing rule. Isn't it? Complementary base pairing rule is going to be applied. Next question number 25. Which of the following statement is not applicable to viruses? Not applicable to viruses. Virus replicate autonomously in the absence of host. It is not applicable to virus. Virus will going to replicate or virus will going to multiply. Whatever the things are there with respect to virus, it want one living host. Living host for that virus. Yes, you are right, Rakesh. For 24th question, C is the right answer. Yes. So for 25th question, D is the right answer. Virus replicate autonomously in the absence of host. It is not applicable to virus. Okay, all other things first A, B, C are right to the question number 25. 26. Mode of DNA replication is what? It is semi-conservative and bidirectional. It is semi-conservative replication, isn't it? So one of the strand from the parent DNA is going to be conserved in the newly synthesized DNA. That is what DNA replication is semi-conservative and bidirectional. In both directions it is going to be happen. That is the replication. 3' to 5', 5' to 3', isn't it? So answer is C for 26th question. Yes. 27. Which enzyme is used to join nicks in the DNA strand? Or we can say that which enzyme is going to be helpful for ligation in the DNA strand? Joining of DNA strand. It is DNA ligase. DNA ligase helps in the joining of DNA strand, isn't it? DNA ligase. Which enzyme is used in the unwinding of DNA? Unwinding of DNA molecule is required during the time of replication and transcription. So which is that enzyme? It is a helicase. Especially in the replication it is must. It is a helicase enzyme, DNA helicase enzyme. So 27. Second, unwinding is uncoiling. So DNA is in coiled format. Uncoiling of DNA is done by helicase enzyme. 28. C is the right answer. And last two questions. 29. Which of the following process does not occur in prokaryotes? Does not occur in prokaryotes. Transcription, splicing, translation, replication. It is nothing but splicing. Splicing is not required because there is no intron-exon concept in the mRNA production under prokaryotes. So splicing phenomenon is not there in the case of prokaryote, but it is present in the eukaryote. But transcription, translation, replication is both observed. All the three steps are observed in the case of prokaryote and eukaryote. So answer is 29th. B is the right answer. Yes, Sheshika, it is right for 29th. And for 28, you are right. That is C is the right answer. And last question, cDNA, complementary DNA. cDNA refers to complementary DNA is synthesized from RNA by the enzyme called reverse transcriptase. Reverse transcriptase, or it is the enzyme which is responsible for the conversion of RNA into DNA or circular DNA. We can say, complementary. It's not complementary, circular DNA. Sorry for that one. So it is circular DNA synthesized from RNA. RNA is going to be converted into DNA, especially observed in the case of viruses like RNA viruses, for example HIV, which is undergoing reverse transcriptase phenomenon. So by this I am going to wind up this class. Okay, good night to all. This is Unacademy Plus subscription slide, Karnataka CET 2021 aspirants for that persons, comprehensive batch and champion batches, then daily T20 free practice test. So thank you one and all. We will going to meet on 23rd. Thank you.